Hello everyone, my name is Tanushree Tunstall and my poster is about structural and genomic insights into pyrazinamide resistance in Mycobacterium tuberculosis. Tuberculosis, despite being an ancient disease, still remains a public health burden, compounded by the rising numbers of drug-resistant TB cases as shown. TB is caused by the bacterium Mycobacterium tuberculosis, or Mtb in short, which has a clonal genome. Drug resistance in Mtb develops through both intrinsic and acquired routes, as indicated. However, due to Mtb's clonality, the primary route by which it acquires resistance is through non-synonymous single nucleotide polymorphisms, or NSNPs. It is also worth remembering that the intrinsic routes can be driven by acquired routes, like for example mutations driving changes in efflux pump activity. Resistance-associated point mutations have been described for nearly all anti-TB drugs. My work focuses on understanding the impact of NSNPs on drug target structure, and hence I'll be using the words mutations, point mutations, SNPs, and NSNPs interchangeably throughout this presentation. Computational tools can estimate the biophysical effect of stability as a change in Gibbs free energy, delta G, giving us insight into how mutations alter stability. Further, these tools can also estimate changes in the binding affinity of ligands upon mutations. So, we use these tools to investigate the effects of NSNPs in pncA on its structure and its interaction with the anti-TB drug pyrazinamide, which is a crucial first-line antibiotic used in current TB treatment. pZA is a prodrug which is activated by the amidase activity of the enzyme pyrazinamidase, encoded by the pncA gene, converting pZA to its active form of pyrazinoic acid. Despite its indispensable status in TB treatment, its exact mechanism of action remains poorly understood. The data in our study came from two sources, genomics and structural. Genome-wide analysis from nearly 35,000 isolates provided the genomics data related to pncA, while the corresponding structural data was obtained from the PDB repository and molecular docking as indicated. Foldex and MCSM computational tools were then used to understand the structural consequences of pncA NSNPs. So we wanted to look into some particular questions, namely impact of NSNPs on pncA, impact of pncA's molecular motion on drug mutations, how these stability measures relate to GWAS measures of allele frequency and odds ratio, and finally, how they relate to Mtb lineages. So, the top figure shows all 424 NSNPs associated sites mapped onto pncA structure, colored by their association with pZA resistance. As you can see that the purple and pink areas surround the pZA binding site, while the blue areas lie further away. This corresponds to the sites that have mutations associated with pZA resistance, denoted as DM, and you can see in the box plot below that indeed statistically drug mutations lie closer to the pZA binding site. Moving across, we see that over 80% of the non-synonymous SNPs have destabilizing effects for both overall stability and ligand affinity. It is likely that this comes without a massive fitness penalty, as pZA is a prodrug. We then move on to the bottom figure, which is a bar plot showing all 424 pncA mutations by site, with the active site positions indicated in color on the horizontal axis. The vertical axis shows the number of mutations at each site. Each bar is a single NSNP, colored by the DUET stability effect, with color intensity corresponding to the extent of their respective effects. As can be seen, 82% sites are associated with NSNPs, with over 50% sites associated with multiple SNPs, which include all active site residues. We then wanted to investigate the differences between drug mutation and other mutations, as shown in the left figure. We found that drug mutations lie closer to pZA binding site, as I mentioned earlier, have lower binding affinity, and interestingly, have higher Foldex delta G compared to MCSM duet delta G. This suggests that the drug mutations display larger local perturbations in the mutated region considered by Foldex compared to the lesser effects of surface area considered by MCSM duet. Moving on, right figure. We also wanted to understand the impact of pncA's molecular motion to see if it helped explain some of the DM-OM distinctions. We used the Dynamute web tool for this and found that the molecular motion in pncA does not interfere with pZA binding, as active site residues were not associated with high fluctuations, with large molecular motions mainly restricted to the loop region 60 to 66. Moving on, In a related study by Potelli et al. 2018, looking at the impact of point mutations on other TB targets, proposed a theoretical bacterial fitness and protein energy plot based on observations that mildly stabilizing mutations confer local fitness advantages and that frequently occurring mutations didn't result in extreme impacts. We therefore wanted to investigate this using our dataset. So, the bottom left graph shows 3 bar plots of NS SNPs on pncA and highlight the prominence of different positions in relation to MAF, OR and frequency of pncA mutations. Part C of the bar plot highlights that active site residues are under drug selection pressures due to the presence of multiple mutations at these sites. Measuring this relationship empirically using Spearman correlations on the right plot, we find that MAF and OR were moderately and significantly correlated, as shown by the blue box. However, there were weak associations between MAF, OR, TUET and FoldX indicated by the red boxes. Finally, we wanted to investigate the relationship between Mtb lineages, stability and mutations associated with pncA resistance. It was noted that while most samples were from lineage 4, it displayed the least mutational diversity, while lineage 1, despite having the fewest samples, displayed a high mutational diversity after lineage 3. Statistically significant differences were observed between lineage 1 and other lineages except 3 and 4. But most interestingly, Mtb lineage 1 exhibited a distinct peak for DM mutations as indicated. This suggests that the ancient lineage 1, consisting of point mutations with extreme biophysical effects, may be rapidly giving way to other modern Mtb lineages linked to MDR and XDR-TB and virulence. I'd like to end by acknowledging my supervisors, Dr. Nicholas Fernham and Prof. Tane Clark, and the lovely postdocs Jody and Charlotte, and to you all for listening. Thank you very much.